Electron microscope. Electron microscopy is used when the greatest resolution is required and when the living state can be ignored. The images produced in an electron microscope reveal the ultrastructure of cells. There are two different types of electron microscope: the transmission electron microscope, TEM, and the scanning electron microscope, SEM. In the TEM, the electrons that pass through the specimen are imaged. In the SEM, electrons that are reflected back from the specimen, secondary electrons, are corrected, and the surfaces of specimens are imaged. The equivalent of the light source in an electron microscope is the electron gun. When a high voltage between 40,000 and 100,000 volts, the accelerating voltage, is passed between the cathode and anode, a tungsten filament emits electrons. The negatively charged electrons pass through a hole in the anode, forming an electron beam. The beam of electrons passes through a stack of electromagnetic lenses, the column. Focusing the electron beam is achieved by changing the voltage across the electromagnetic lenses. When the electron beam passes through the specimen, some of the electrons are scattered while others are focused by the projector lens into a phosphorescent screen or recorded using photographic film or a digital camera. The electrons have limited penetration power, which means that specimen must be thin, 50 to 100 nanometers, to allow them to pass through. Thicker specimens can be viewed by using a higher accelerating voltage, for example in the high voltage electron microscope, HVEM, which uses 1 million volts of accelerating voltage, or in the intermediate voltage electron microscope, IVEM, which uses an accelerating voltage of around 400,000 volts. Here, stereo images are made by collecting two images at 8 to 10 degree angles. Such images are useful in assessing the 3D relationships of organelles within cells when viewed with a stereoscope or with a digital projection system. Contrast in the EM depends on atomic number. The higher the atomic number, the greater the scattering and the contrast. Thus, heavy metals are used to add contrast in the EM, for example, uranium, lead, and osmium. All of the water has to be removed from any biological specimen before it can be imaged in the EM. This is because the electron beam can only be produced and focused in a vacuum. The major drawback of EM observation of biological specimens, therefore, is the non-physiological conditions necessary for their observation. Nevertheless, the improved resolution afforded by the EM has provided much information about biological structures and biochemical events within the cells that could not have been collected using any other microscopical technique. Extensive specimen preparation is required for EM analysis, and for this reason there can be issues of interpreting the image because of artifacts from specimen preparation. For example, specimens have been traditionally prepared for the TEM by fixation in glutaraldehyde to cross-link proteins followed by osmium tetroxide to fix and stain lipid membranes. This is followed by dehydration in a series of alcohols to remove water, and then embedding in a plastic such as EPON for thin sectioning. Small pieces of the embedded tissue are mounted and scrubbed on an ultramicrotome using either a glass or diamond knife. Ultra-thin sections are cut to a thickness of approximately 69 nanometers. The ribbons of sections are floated into the surface of water and their interface colors are used to address their thickness. The desired 60 nanometer section thickness has a silver-gold interference color on the water surface. The sections are then mounted into copper or gold EM grids and are subsequently stained with heavy metals, for example, uranyl acetate and lead citrate. For the SEM, samples are fixed in glutaraldehyde, dehydrated through a series of solvents, and dried completely either in air or by critical point drying. This method removes all the water from the specimen instantly and avoids surface tension in the drying process, thereby avoiding artifacts of drying. The specimens are then mounted into a special metal holder or stub and coated with a thin layer of gold before viewing in the SEM. Surface can also be viewed in the TEM using either negative stains or carbon replicas of air-dried specimens. Immuno-EM methods allow the localization of molecules within the cellular microenvironment for TEM and on the cell surface for SEM. Cells are prepared in a similar way to indirect immunofluorescence, with the exception that rather than a fluorescent probe bound to the secondary antibody, electron-dense colloidal gold particles, 10 nanometers, are used. Multiple labeling can be achieved using different sizes of gold particles attached to antibodies to the proteins of interest. The method depends upon the binding of protein A to the gold particles, since protein A binds in turn to antibody fragments. Certain resins, for example, low-acryl and LR white, have been formulated to add antibodies and gold particles to be attached to ultrasound sections for immunolabeling. New methods of fixation continue to be developed in an attempt to avoid the artifacts of specimen preparation and to observe the specimen more closely to its living state. Specimens are rapidly frozen in milliseconds by high-pressure freezing. Under these conditions, the biochemical state of the cells is more likely to be preserved. Many of these frozen hydrated samples can be observed directly in the EM, or they can be chemically fixed using freeze substitution methods. Here, fixatives are infused into the preparation at low temperature, after which the specimen is slowly warmed to room temperature. Using cryo-electron tomography, cryo-ET, the 3D structure of cells and macromolecules can be visualized at 5 to 8 nanometer resolution. Cells are typically rapidly frozen, fixed by freeze substitution, and embedded in epoxy resin. Thick 200 nanometer sections are cut and imaged in the TEM equipped with a tilting stage. A typical tilt series of 100 or so images is collected in a digital form and exported to a computer reconstruction program for analysis. By using electron tomography, a 2D digital EM image is converted into a high-resolution 3D representation of the specimen. The method is especially useful for imaging the fine connections within cells, especially the cytoskeleton and nuclear pores, and elucidating the surface structures of viruses. The same specimen can be viewed in the light microscope and subsequently in the EM. This approach is called integrated microscopy. The correlation of images of the same cell connected using the high temporal resolution of the light microscope and the high spatial resolution of the EM gives additional information by imaging using the two techniques separately. The integrated approach also addresses the problem of artifacts. Probes are now available that are fluorescent in the light microscope and are electron dense in the EM.